Yeah, that was good question. I literally forgot everything. I don't know how that
Today. It's this evening at 8 o'clock. The discussion boards will pure magically Iroquois. Okay, if you don't mind, I'm going to try to get going. I know a lot of you have been interested in the neuroanatomy marks. Hopefully you all did well in your neuroanatomy exam. Um, Also, it's a really great time to be in neuroscience, as you know, or if you have been reading any of the local media or the U of T media, you know that one of our colleagues, Dr. Graham Collingridge, actually won the Brain Prize, which is a $1 million award. And actually, Graham Collingridge is one of those individuals who's been involved in LTP, synaptic plasticity for a while. And he actually worked with one of the first individuals that came up in 1973 with the model for synaptic plasticity that we use still to this day. This is Trevor Bliss, along with Turge Lomo, and he's actually worked with those individuals. So it's like almost seeing someone who has worked with one of the classic neuroscientists. So if you have an opportunity in physiology to go see him, please do so. He's a great individual, knows a lot about LTP, synaptic plasticity. I've worked with him in the past, and he did a sabbatical in our lab previously, so he's a fantastic resource, and I think we're really lucky to have him here at the University of Toronto. I'm going to tell you a little bit more about schizophrenia. As I told you, this is actually a really great paper. The paper itself is fantastic in the way that it sets up GWAS. I want you by the end of this course to at least understand some of the things that we have to take into account when you do a GWAS study. It's the same thing that you'll need to be able to demonstrate for your final exam as well. You'll need to demonstrate that you know what sorts of things that you have to put into a GWAS. You'll be given a scenario where you will maybe think about designing an experiment related to a GWAS. And I think as long as you followed what happened in the last lecture, today's lecture will sort of highlight some of those challenges that we have. And I mentioned to you that schizophrenia, we've known about it for a very long period of time. This is one of those papers that brings together a lot of different elements. It is not a perfect paper though. And I'm going to show you one of the elements where this paper, even though it's fantastic, actually breaks down a little bit. And as I was mentioning to some of your colleagues just before the start of class, if you're thinking about how to design an experiment that does a future grant, this is one of those types of papers that you want to read to see what you would want to do next. Their future experiments are actually left hanging, even though they do functional studies, which is a characteristic feature of GWAS. In GWAS, it's not enough to just show that you have a SNP or an RS something 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 that is associated with schizophrenics. You also have to demonstrate functionality. Now, I'm going to ask you at the end if you think that this functionality study that involves this complement factor 4 or complement component factor 4 is actually all that impressive. I'm going to argue with you that perhaps it isn't. Their functional studies were a little bit lacking in this particular case, even though it is now considered a classic paper. So I'm going to go back and talk to you a little bit more about these haplotype blocks. I sort of left things off with the fact that using previous papers that were done by Kim McAllister back in 2012, they went back and revisited the same chromosome, the same haplotype blocks. They tried to get a good idea as to what was going on there in schizophrenic individuals, and they came up with this C4 structural allele that maybe people had missed before, because there are so many different variants of C4, maybe people had missed that. So this is that GWAS study that I mentioned to you at the end of the last lecture. We looked at the Bonferroni correction. This is the Bonferroni correction. There are many, many SNPs that are also found in schizophrenics versus normal individuals. However, if we look at the original Kim McAllister on chromosome, on this particular chromosome number 6, and we take a look at the entire length, this is that one SNP that was found by Kim McAllister previously. So they're identifying something that was previously known in their own studies. They are referencing something that has been published in the literature, and this is that value. So it meets one of the criteria that I mentioned to you earlier for any type of SNP studies. And this is the SNP that they're identifying on the same chromosome that has been identified and associated with schizophrenia before. And this is the C4 expression. And compared to C4b, C4a is much, much higher in terms of its statistical probability of being associated with schizophrenia. The predicted C4b expression is way down here compared to the C4a. So there's a lot of evidence on this GWAS study that involves tens of thousands of individuals that perhaps C4a is indeed something that is associated, that is inherited along with some of the other things that are involved in schizophrenia. Now, lots of things are being inherited or are also found in schizophrenics. Many, many SNPs are being found here. All of these individual points show that there is a really high R² value associated with all of these other SNPs too. How do we know that this SNP over here isn't really responsible for schizophrenia? We have to go back and do some functional study. Sense. Now, this is where I sort of left off. If we go back, we had 4 different types of C4 structures that were identified in this paper. So we had short forms and we had long forms. Some of those involved insertion, the dark gray hatched line here, of the human endogenous retrovirus that overall increased expression. And we have long forms of the C4A or AL, and then we have the C4B, the short form, or the C4B long form. If we were to take a look overall and we looked at the brain cRNA levels of these different individuals, and we went back and we tried to map in individuals that have schizophrenia, when we look at the RNA levels in individuals with schizophrenia, what is the overall likelihood that individuals with schizophrenia are going to have the AL or the C4A long form that causes schizophrenia, etc. So if we take a look, one of the things that pops out almost immediately, the overall risk or odds ratio that is associated with having the C4A long form actually is significantly increased with schizophrenia, especially if you have two copies of this. If you have one from your mother, one from your father, and you have the C4A long version, then the overall risk associated with this, or the odds ratio, is about 1.3 times higher, almost 1.3 times higher than you would have it with just the short form. Okay, so again, one of the other things that is really good about this paper, they had mentioned before that there are copy number variations associated with this as well. So you can have as many as 4 different copies, 3 copies, etc. And if you go back and look at the C4A in particular, if you normalize it for the number of chromosomes that might express this, or you look at copy number in other words, when you go back and take a look at this, it is very, very clear that the C4A does something. C4A really increases the risk if you normalize, as is shown down here, the beta value that you have here is the normalized value. So if you have like 2 copies, one from your mom, one from your dad, of the C4a long allele, you have a very, very high likelihood or a high beta value that you will have schizophrenia. So again, very, very clear evidence that something is going on with the C4a variant of this complement component factor. Now, they're not really sure what this is doing right now. All they're doing is verifying that in human tissue that this indeed plays a major role. So again, if you take a look, they do some more types of controls here. This odds ratio, if you take a look at this value down here, this is sort of the average sum value of the major isoforms, how many different combinations are possible. There are more combinations possible, for example, of the ALB, BL forms. But overall, if you take a look, this is sort of the odds ratio that I had presented for you earlier. So what this suggests is that for a given C4 structure, in particular the C4a structure, it doesn't matter what haplotype block it's associated with, it is more likely that you are going to have some sort of increased risk of developing schizophrenia if you have this C4a. And if you take a look at all of the 13 different possible combinations that involve AL compared to the control that they use here, you are at increased risk. So you're at roughly 1.15 times the risk, 1.2 times the risk, or almost 1.3 times the risk with these different C4 structure combinations. So what this means is that regardless of the haplotype that is being inherited along with it, C4a seems to be a really, really strong predictor right now that you will develop schizophrenia, or it's associated with schizophrenia. There's no mechanism at play right now. So what is C4? And I think I had asked you this earlier. I think many of you have already taken physiology, so you might probably have some idea what the complement factor C4 is, and I'll get into that in a moment. So again, one of the other things that you have to be able to show is there actually in the brains of schizophrenic individuals, what is the C4a RNA specifically looking like in individuals who have had schizophrenia? You take out different regions of their brain, and if you read through the entire paper, they actually looked at 5 different brain regions, all 5 different brain regions, cortex, cerebellum, other areas in the midbrain, all of them had increased C4 RNA levels throughout. So 35 schizophrenic individuals versus 70 individuals. This is sort of the modern way in which we plot things out nowadays in terms of showing you actual data instead of giving you just bar graphs. We can see that there is a significant increase in shift in the entire population, except for a few outliers here of the C4a expression in schizophrenia. So yes, C4a is indeed enhanced, elevated in schizophrenic brains. Throughout the entire brain, actually, cerebellum, cortex, and different tissue types. And again, it's about 1.4 times higher, which by non-parametric statistical analysis is very, very significant. So this gets back to what is C4a, and has C4a been examined previously in the CNS? It has. So many individuals have actually looked at C4 and C3 as part of this complement cascade within the brain. And so this is what a complement your blood, part of your, and throughout your blood, as part of the complement system, you have all of these different factors that are there until it makes contact with a pathogen. So when a pathogen comes in contact in your blood with any of these different elements, then this complement cascade becomes activated. And again, I don't want you to memorize this entire cascade. In the immune system, the C4 activation and the C4a activation leads to things like inflammation, but we're not going to try to memorize this entire pathway. It's just there in case you've forgotten that we have a whole host of blood-borne, serum-borne, tissue-specific forms of innate immunity that will recognize and help to eliminate any pathogens that it happens to encounter. And it's there to help to opsonize or to remove any of those pathogens that it encounters that shouldn't be there. So if you have a bacteria or you have something that is part of a bacterial wall, this opsonization process where C3 binds, then activates other complement proteins will then lead to things like opsonization or the inflammatory response, etc. However, in the brain, when individuals went back and took a look at in the brain what is going on here, because the brain is one of those specific compartments that tends to be immune privileged. It doesn't really have the same sorts of immune cells floating around. It has those microglial cells, an immune privileged organ, and in the brain, these genes that are part of the traditional immune response, the classical complement cascade, have actually been implicated in other things. So they're doing other things in the brain. And one of the things that the C4 and C3 systems in particular have been implicated in is the elimination or pruning of synapses. Okay? Is that okay for everyone? So totally different functions, same protein, but where it's in the nervous system, it is doing something a little different than it does, or maybe not so different. If you think about eliminating pathogenic signals, maybe it's not so different in terms of the pruning or elimination of synapses. Do you guys remember anything about schizophrenia at all? What happens to the cortex in individuals with schizophrenia? So there's lower activity, there's that hypofrontality, that's a functional consequence. What else is going on in the schizophrenic brain? If you take a look at the pathology. Yeah, Danny? Yes, you could have increases in some ventricles, yes. But what does that actually imply? Sorry, it could. There's a lot of controversy. Is there more death in schizophrenia or not? What else is happening? Yes, Sally? Yeah, what does cortical thinning mean though? Like the only, thinning of hair means one thing. Cortical thinning means what? What does that actually mean? Yeah. Could represent white matter loss. So one of the things that we don't know is, for sure, it's been demonstrated time and time again that when you look at structural studies or you look at pathology, there is cortical thinning. The cortex itself looks like it is thinned out. It's not as thick as in a normal individual. Now, the tree looks a lot thinner than it does in the summer because in the summer it has all of those leaves. Now, has it grown more branches? Maybe it has, or maybe it will by the spring. But the tree looks thicker and fuller mainly because of the number of leaves that it has. And if you equate the number of leaves with synapses, that way, in the schizophrenic brain, there's been an issue with not necessarily neuronal numbers or neuronal death, again, very controversial, but the cortical thinning might be representing a loss of synapses. Somehow, the number of synapses that we have are just not there. So again, there's a lot in this particular paper, and if you take a look, and I showed you this at the last class, this paper was actually submitted fairly early, and it took them about 6 months for them to actually get it accepted within Nature. And I can guarantee you that they were asked to include a lot of controls. And some of these controls were done sort of haphazardly, and I'll get to that in a moment. This is actually what you'd want to do. So where is C4 in the brain? You have to know this. Other people have investigated this before, but you have to know this. So on brain cells, human brain. So in the human hippocampus So the gray matter makes sense. You know, you have cell bodies that you'd expect to see cell bodies that are C4 positive and mu N positive, but you also see these in the white matter as well. And the highest number actually of C4 positive cells was detected in the hippocampus. And we know that there are issues with memory, attention, cognition, etc., through the hippocampus as well. Interestingly, they also report, they don't actually show this in any of their figures, it's only in their extended data figures, but they also report that they see C4 positive cells, which are new and negative, that are on a subset of astrocytes. They don't identify those astrocytes. So for example, if you thought astrocytes were really important in schizophrenia, because it's just a report, you would probably want to follow up on this in a grant. You would come up with a hypothesis, take their original findings, and then extend them into your hypothesis. Maybe astrocytes are important in the pathogenesis of schizophrenia. Maybe you would want to go back and manipulate these astrocytes to see what's going on in schizophrenia. Eddie, did you have a question? So if they're nu N negative, meaning that they're not neuronal, so nu N is a marker for neurons in this particular case, and they didn't stain for GFAP, or I didn't show you the staining for GFAP, or the glial fibrillary acidic protein, but that's how they came to the conclusion. It's not in nu N, therefore it's not a neuron. When you go back and stain for a glial cell marker, it turns out to be an astrocyte. I mean, there are a limited number of astrocytic markers like vimentin, GFAP, A2B5, and that you can tell the lineage of the different types of non-neuronal cells. Okay, so that might be something that when you're reading your papers, you can take a look at. So if there's a preliminary report on something, for sure, we want to look at neurons, but they were also finding this on a subset of astrocytes. And again, they don't actually show you all the papers here. Now, what makes this also really interesting is when they start looking at the staining, most of the staining that they observed was punctate. So it's not like diffuse across the entire cytoplasm, which tells you one thing. It's found in a punctate pattern. What does a punctate pattern represent for you? Or what do you think it means in a neuron? And let's assume that we're talking about neurons. Punctate. Yep, Julian? Could be synapses. We don't really know. And that's one of the things that you have to be able to go back and check out. So we see these little speckles, these little dots that you see here. And again, they use a couple of really advanced techniques. So I told you about the digital droplet PCR. One of the other upcoming techniques that are being used now, especially when you have a section of the brain. So the brain has many, many synapses, many, many neurons packed together in a small area. In order for you to be able to get a good signal, you have to have some way to resolve all of that fluorescence. And one of the ways that we can do this is using a new microscopy technique known as high resolution structured illumination microscopy, or SIM. And that's particularly useful when you have a dense network of cells that are put together. And this high resolution imaging that you see here on panel A and panel B allows you to see individual puncta, even if you have like lots of puncta in a very, very small space. And particularly in the hippocampal formation, which is being highlighted for you in B, when you go back and take a look at all of the different markers that are being expressed, so they tried to do a quadruple stain. So this was actually not something that you see a lot of. Usually you see like 3 stains, or you see 2 stains at a time. So they did 3 over here. But actually here they're looking at C4 in green. They're looking at PSD in red. They're looking at the glutamate vesicular transport protein, VGLUT1 and 2 in hooks. And one of the things that they see is that it localizes with both the pre-synaptic component as well as the postsynaptic component. They see it in both places. Again, which means that it's probably associated, maybe, if it's associated with, for example, PSD95 is a postsynaptic marker. So when they see these co-localizing with PSD95, it has to be postsynaptic. That's the only way that it would be represented. Or if they see it also co-localizing with the V-glute presynaptically, then that also suggests that it's presynaptic. So they are seeing co-localization with these pre as well as postsynaptic marker proteins. So in other words, they are parts of synapses. They are involved in synapses, in other words. Any questions about that? And is PSD95 something that you have all heard about before? Can anyone tell me really quickly what PSD95 is? No? Right? Yeah, so the postsynaptic density protein runs at about 95 kilodalton, so we call it PSD95. It's a good marker for excitatory postsynaptic neurons, which is why we look at V-glut1 and 2, which is a marker for glutamate transporters to package glutamate into their neurotransmitter vesicles. So again, I don't know how much background all of you have in neuro, but to me this was actually one of the more crazy figures. How many of you have worked with cell culture? A lot of you have, good. How many of you have worked with primary cell cultures? Okay, primary cell cultures, Priscilla, in what? Like what kind? Microglia and astrocytes from what animal source? Rats, okay. Have you ever grown neurons from rats or mice? So growing rat and mice primary cultures is challenging, but they're actually growing primary human cortical neurons, grow them in a culture dish, which is what you're seeing here, because neuronal cultures are not like the human brain where you have them all densely packed together. They don't need to use this structured illumination microscopy technique. They can use just normal confocal scanning microscopy, which is why you're seeing this here. And these confocal images of the primary human cortical neurons shows that the C4 is co-localizing with MAP2, which is a dendritic marker. It's also found along neuronal processes. And basically what they're showing is, just like they did in the human cultured brain, in the human sectioned brain, they're showing exactly the same sorts of patterns, that we see C4 on a more precise level using confocal microscopy at the synapses. So we see it in PSD95 positive processes. We see this with the presynaptic marker synaptotagmin, and we also see it associated with PSD95. So it is synaptic. It's synaptic in a human preparation, both from slices taken through a human brain with the previous image, as well as in primary cortical cultures. So it's pretty solid evidence that C4 does exist in synapses in a human preparation. It's not in mouse, it's not in rat, it's in a human preparation. And again, for them to be able to grow human cultures is pretty impressive, at least for me. So this shows you that through their GWAS study, they are able to demonstrate that there is clustering and association of the RNA based on their GWAS studies with schizophrenia, and especially the C4 isoforms. It also shows that at the level of the synapses, using confocal and SIM microscopy, that it's definitely found at the level of synapses in a normal human brain. But then you have to wonder, so what? So is this really a good functional study? At this point, it's just a descriptive study. There's no function associated with it. So the first thing that you want to try to do is to make some sense out of what you've observed. Also observe that there are complement proteins that exist in the human brain. So I'll just draw your attention to this, because you're going to have to tell me at the end if you think that their experiments actually resolve this issue or not. So far, this is like a fantastic paper. They're setting in place the GWAS study, which then leads to the RNA studies, which then lead to the protein studies, all indicating that C4 is found at the level of the synapse, increased. I'm hoping that you're all with me so far, right? So this increased association of C4 with schizophrenia, also finding C4 in human synapses. Other studies have shown that C3, other complement proteins, are also found in synapses. We know that there's an issue in schizophrenia associated with synapses as well, or potentially cortical thinning associated with synapses, and maybe in synapse elimination. And other reports have suggested that when you take a look at the the pathology of individuals with schizophrenia, that there is a decrease in the number of synapses that are associated with individuals that have schizophrenia. So it suggests that somehow C4 might work with some of the other components, like the others have suggested, to affect synapse elimination. Are you with me so far? Does that sort of make sense? Is that rationale somewhat sound? Do you agree? Okay. I mean, this is Steve McCarroll, who's been working on schizophrenia for a long time. He's one of our best neurogeneticists, so it's really hard to argue with the logic here. So functionally, how do we go back and test this? Now, this is where you have to think a little bit. So one of the things that they used was a mouse model. Now, this mouse model has previously been described. This mouse model exists. It's in the literature. You don't have to create it de novo. Now, remember the timeline. He had submitted this for publication back in May. It got accepted in December. And so he was probably making use of whatever models were available to him. A couple of things to keep in mind. Within the mouse genome, even though our genomes are relatively similar, in the mouse genome, a C4 gene actually looks like both C4A and C4B. There is no difference. It only exists, by the way, in humans where we have C4A and C4B. In other primates, we also don't see this differentiation of C4A and C4B as readily. So as I mentioned to you before, in the immune system, the C4 promotes some C3 activation, et cetera, which then allow for the opsonization, the engulfment, and then removal by phagocytic cells of whatever pathogenic targets are found within your bloodstream or in your tissues. However, in the development of the mouse brain, C3 actually seems to be associated or found with a number of different synapses. And we know that C3 is required for synapse elimination. The move for myocytes within the these receptors. Let me just try to get that laser pointer back. So here's where I need you to think a little bit. And again, I'll ask you this in a few minutes. So these authors know that there's an association with C4 and the complement system. We know that the complement system, the C3 system, has been implicated previously in the elimination of different synapses by microglia when that complement system gets activated. So what these authors decided to do was to use a mouse that is deficient in C4. So do you think that's a good idea? Yes?
If there's no distinction between C4A and C4B,
Yep.
and C4B, like the two, like being BB, makes you at no greater risk,
Yep.
then wouldn't removing C4 completely in mice not be the most accurate representation of what it'd be like in humans?
So for those of you at the back, is this an accurate representation in humans? If you're eliminating or removing both copies of C4A and C4B, is that what we were looking at in schizophrenia? In schizophrenia, when you look at those postmortem samples and you look at RNA, actually we saw an increase in C4A levels. They're looking at what happens when no C4 is there at all. So what they're just trying to do is show you that there is a functional consequence of when C4 and C3 don't interact. They're not showing you what happens when C4 it gets overexpressed, which is what the proper experiment would have been. That C4 and C3 in normal development are actually important, and here are the consequences of it. Now, ideally, they should have done the C4A overexpression experiment, but they didn't have time. After it was submitted in May of 2015 to get it published by by December of 2015, that 6-month gap or whatever it is, these types of experiments take a lot of time for the mice to be bred to the right age, for the staining to be done, and the quantitation. This is probably why they use a deficient mouse. But this gives you a wonderful opportunity, if you were to do this sort of as a future grant proposal, to do the experiment that they didn't. Like, what would happen if you overexpressed it? So before I get into your grant proposals, etc., so this opportunity exists if you're writing a grant on something to look at the experiments that were done and what gaps are there in the experiment. I've already addressed one gap with the astrocytes, and this might be another one. Instead of overexpressing, they have a knockout or a deficiency in C4. So what they saw was that when you knock out C4 and you go back in the C4 mouse and you take a look at the expression of what it normally binds to, the C3 immunostaining in an area of the brain. Do you guys know what DLGN is? Yes? What is it, Danny?
Dorsal lateral geniculate nucleus?
Sure. So it's the dorsal lateral geniculate nucleus, which is associated with what? Lateral geniculate, pathfinding. It's involved in vision, right? So when you're looking at the movement from the retina up into the visual cortex, the lateral geniculate nucleus and the order of which the synapses occur within the lateral geniculate nucleus are really important. So this is an important structure to look at development and normal synaptogenesis. So the dorsal lateral geniculate nucleus, which is an important developmental area, they found that there was a reduction in the C3 staining compared to the wild-type littermates. Also, there were fewer synaptic inputs that were C3 positive when you didn't have in the knockouts any of the C4 around. So what it really suggests is that you need normal interactions between C3 and C4. Again, nothing earth-shattering. It's just a sort of validation that, you know what, normally C3 and C4 interact, and this is what they're trying to show. And again, it's very, very conclusive. There's no doubt. I mean, if you just take a look at this one panel here at the top, when you're staining for C3 on the top in a wild-type animal, and the same area of the brain in the dorsal lateral geniculate, the C3 staining in a C4 knockout is almost gone. It's almost completely eliminated. It's black. And so when you go back and you take a look, it's not surprising that the immunoreactivity that you're measuring from the top panel here to the bottom panel here between the wild-type animal here and the C4 knockout animal down here, it's not a huge surprise that the C3 immunostaining that you see down here is almost eliminated. They're normally supposed to interact. When the interaction is now gone, C3 also disappears. Surprisingly, if you take a look, the density of some of the other proteins remains more or less the same. So it might mean a lot of different things. I won't go into it in a lot of detail. The staining itself is pretty conclusive, at least on this, but their experiment itself is not very impressive. Any questions on this before I go on? And you guys kind of remember the lateral geniculate, the reticulogeniculate system, the pathfinding from the retina back to lateral geniculate before it gets into the primary visual cortex? Kind of. It's not that important. You're not going to be tested. It's more for my sort of information. And again, some really great high quality immunoscans that they did. You'll just have to take my word for it that their immunostaining is like really first rate, amazing resolution that they're getting on their punctate patterns of staining. So they're not really sure what all of human brain. Normally it's expressed by neurons. Normally it gets expressed and transported out to dendrites, maybe axons. We saw that with the neurofilament stain. Definitely gets into synaptic regions. It gets there, and we know that that's there in PSD positive postsynaptic regions. It may also be secreted because we saw the punctate localization with the V-glue transporters. So it's all over the nervous system. And again, in mice, one of the things that C4 is normally supposed to do is to promote synapse elimination in a developmentally timed series. So especially in the visual cortex, which is what they looked at, you would normally see or have C4 causing synapse illumination. They actually didn't show that in this particular paper. But this is sort of the literature that's associated with C4. And what they are looking at is that C4 knockouts have a lack of C3, but they actually don't show this synaptic elimination. But it would make sense. If you have like too much C4, maybe you eliminate more synapses. They didn't show that, but that's sort of the implied message from all of this, that normally C4 is supposed to interact with C3 elimination, C4, which they didn't do. Maybe you overactivate this system and cause more synaptic pruning or elimination. Again, it would have been a great thing for them to do, and I'm guessing that if, as a follow-up, that's indeed what they may try to do. Danny?
Were C3 levels normal for both the control and wild types? I mean, the idea is that C4, if C3 is more responsible for synaptic pruning with the help of C4, could
Yeah.
C3 be higher or lower? Could it just?
So in their GWAS study, for example,
It was just C4.
yeah, they were only looking at C4. It's a good question. I can't remember in their extended data if they actually look at C3 levels. I don't think they were changed, and I think that other individuals that have looked at that cascade system really didn't see any change. Their main thing was they see differences in the transcript of C4A, and then they kind of went into just general C4. So again, in humans, because it's a developmentally regulated change in the number of synapses and the elimination of synapses, if indeed C4 is being overexpressed in schizophrenia, it might lead to changes in synaptic pruning, cortical thinning, etc. And the synapse elimination in different areas, like the association cortices, the ones that are outside of the main areas or the main cortices, like the visual cortex or the motor cortex, the associated cortex or the sensory cortexes, these association regions actually get pruned during adolescence all the way into your 30s. And this is sort of the age range at which we start to see schizophrenia manifest itself. So it's a possibility. It's all correlational. It's all actually just pie in the sky at this point. But it makes a very intriguing story. There are lots of things that, for example, you would use this paper to go and investigate next. You might want to investigate point number 2. You might want to say, well, you know, I want to actually see if critical time period of 18 to 30 is actually going to regulate synaptic elimination. And I might try to do that in a mouse. So at the end, their discussion, by the way, is really, really short for a Nature paper, basically because they had nothing really much to say. And this is sort of the encapsulation of their Nature paper discussion. So these two C4 genes, both C4A and C4B, have a really strong correlation through GWAS studies with the risk of schizophrenia, and especially that C4A isoform that we talked about before. Now, based on other biochemical studies, it turns out that the C4A, they don't follow up on this. This is from literature. They don't actually have proof that this occurs in their own hands. But in the literature, C4A seems to form bonds more readily with proteins, and C4B seems to bind more favorably with carbohydrate surfaces. So is it theoretically possible that C4A does indeed have more protein-protein interactions as with like C3? Perhaps. Again, it's all theoretical. It's only in their discussion, and it suggests that maybe if C4 is normally more likely to interact with proteins, other proteins, including C3, that maybe if you overexpress C4A, that somehow by binding to something, maybe C3 at the level of the synapse, it might cause their elimination. How all of this works, they're not 100% sure. Now, one of those caveats that I had for you earlier was that in a GWAS study, you have to have some sort of correction for statistics. They definitely have that. They have included previous data as a criteria, so they definitely have that. I'm going to ask you, do you think that their functional data that they presented here is convincing? So have they actually shown you that this is indeed the SNP that causes schizophrenia? Agree, disagree, not sure. Maybe? Disagree? Do you think they have a compelling case? Eddie, do you have an idea? Do you agree with this is a good functional study? It's good enough for a Nature paper. Yeah, for sure. Yeah, it's definitely something they probably. Yep, I would agree with you. Yeah. Yeah, the joke is it's good enough for Nature, not good enough for Cell. But maybe that's something different. So the other thing that is really appealing about this paper, as Eddie was saying, this fits with everything else in the literature, like really, really well. It dovetails really well. Kim McAllister's 2012 paper, another landmark paper, saying that, you know what, we don't know and we don't understand this either, but it's not anything to do with dopamine, because even here in Toronto, we have about 6 or 7 different primary researchers dealing with dopamine receptors, changes in dopamine binding proteins. It makes sense. Dopamine has been implicated in schizophrenia. But the reality is, when Kim McAllister came up with her 2012 paper that implicated something on chromosome 6, which no one was able to map because that 100 megabases or so on chromosome 6 that involved the major histocompatibility complex was really too big to map out at the time. This all dovetails perfectly with that. There's an area in there. It's hard to detect it because there are 4 different structural variants that also have like different copy number variations. There's an endogenous retrovirus that's stuck in there as well. So this dovetails really well. It's found in the synapses, but I also agree it's not probably at the functional level just yet. Someone will do it, but not right yet. Yeah, Richard? Yeah. No, I mean, in terms of the visual studies, it wouldn't make a lot of sense for them to do a knockout and then it would make more sense to do the visual overexpression and then see that. But for sure, I think that would be, for me, if you were to suggest as a follow-up to this particular paper that you wanted to do a functional study and then you outlined the functional studies that you might use, and even better, one step further, also have a known non-genetic schizophrenic model that you would compare to, then that would be like, for me, an A+ sort of grant proposal. Any other suggestions, ideas before I remind you of a couple of things today? So, okay, yeah. Yeah. Yeah, so that's a good point. So they don't see tremendous differences in the number of synapses, which is why the rest of the staining, the ones to the right of that figure, actually didn't look all that different. That's actually been a bone of contention for a lot of individuals. So if you knock out C3 or C4, you should see lots of synaptic changes. It doesn't seem to be that consistent. So it's a good point. Okay, so a couple of things. So this evening, on your discussion board, so it'll be as 3 separate links. You'll have to sign up for one of the different dates. There'll be a discussion board thread for March the 17th, another one for March the 24th, and one for March the 31st. One person from your group should be signing up for one of those different dates. And we'll take up to 6 groups per date. You'll be split into 2 different groups. So there'll be 3 presentations on Thursday in one room. There'll be 3 presentations in another room on the 17th, etc., for the 24th, as well as the 31st. If you have issues with not being able to attend because of work or other things, just let me know. I'm happy to try to accommodate you. It'd be nice and it'd be really professional if you can show up, and I will allot just for showing up marks associated with attendance. Your written review is due on March the 8th at the start of class. So I think the template is out there for you. Some exemplars are out there for you. The other thing that you can do if you're a little bit concerned about this is that I think, as I recall, I asked this up so that you can actually submit and see your originality score for Turnitin before you do your final submission, just to make sure that everyone's feeling comfortable with it. And the last thing, if you're really worried about the quality of your writing, you can also share that as a Google Doc with me over the weekend, and then I can take a quick look at it and give you some comments back before you do your submission. Okay, so just keep in mind tonight's deadline. Make sure that you're on track. Make sure that you're in contact and communication with your group. And I'll see you all on Tuesday when we start moving into prion diseases.